Could you start the recording? Thanks very much, Risa. So today's going to be focused on protocol metadata. Yesterday, we had a really good discussion around the work we've done so far on making protocol templates, and then also talking about other efforts for protocol standardization and sharing. And so I thought today we'd really focus in on our discussion around MIOP, or minimum information for an omics protocol. So to start out with, I'm just going to give a short presentation going over again what work we've done and then focusing in on MIOP. So let me share my screen. All right. So I thought I'd start off again with saying, introducing Bebop as a project. So a lot of this work got started in 2020 through an OBPS workshop where a group of us came together and formed an Omics and eDNA task force, where we saw a lot of needs for how to handle different omic protocols. And this has now evolved into a UN Ocean Decade project called Better Biomolecular Ocean Practices, or Bebop. And we're organized under the OBON program. So OBON stands for Ocean Biomolecular Observing Network. And its mission is really to create this multi-omics diversity observing system and connect different projects that are conducting omics observations, and also to develop and transfer capacity to create additional observational capacity, and then use omics data to enhance marine ecosystem models and address scientific management and policy questions. So Bebop is kind of sitting under this larger UN Ocean Decade program. And so just to talk about some of the work that we've done since 2020, as a group, we came together and tried to think about a better infrastructure for sharing omics protocols. And so one key idea we came up with was splitting up a study across protocols for different components. So here, this is a meta-barcoding pipeline where you have your experimental design, your permits that you had to get for actual field sampling, and then all your lab work, bioinformatics, data analysis, and releasing the data. And so we kind of came up with these different methodological categories that describe the different kinds of protocols that go into an omic study, here with meta-barcoding just as an example. And so here, each of these boxes represents a different protocol. And we've made protocol templates that can standardize how we compare these different types of protocols. And so one key part of this too is we decided there's some key metadata that we need to describe each protocol that lets us compare them as well. So the first metadata term is methodology category. So this is just referring to those categories I presented in the last slide, from sampling design all the way down to here, I can go back. Sampling design or omics sequencing procedure to data analysis. So these kind of broader thematic categories of different kinds of protocols. So that term will go under methodology category. And then we have different terms that describe the project the protocol is designed for, the purpose of the protocol, resources and analyses, terms that describe where the protocol was designed to be applied, and then also what kind of medium you're sampling and what target, what's your target, if it's a taxa, gene, or molecule. And so we started out with this set of terms, and we created a GitHub repo to discuss and track changes as these terms evolve. And so, we've done these for the MIOP terms, and also yesterday I talked more about the protocol templates, which, as an example, this is an example of the PCR protocol template we came up with, but it's just a way of formatting protocols into a similar format so you can compare them easily and track their changes through GitHub. So with Bebop, we're trying to use these two functions, these protocol templates and this MIOP data, to map the protocols that are being used across the OBON project. So we really want to use these OBON projects that are as like a test case for how we can compare protocols across these different omics sampling programs. So here again are the initial MyOp terms that we defined in our paper, but we've made some changes since then. And we're tracking those changes in a YAML document. And I thought I'd just share an example real quick. We're not going to be editing this document, but if you go onto the GitHub site, this is how we're storing the different terms. And so once you get a hand of how to read this, it's pretty simple. We just have the name of our term, we give a definition of the term, some examples, and then some key things that the YAML file uses to keep track of what exactly the term is. So when we're making edits or requesting changes on GitHub, it's to this terms.yaml file. And the nice thing about this is also how the Genomic Standards Consortium keeps track of their MIXS standard. So we're hoping to incorporate our standard that we develop into the GSC's standards. So some of the changes we've made is we've decided to remove this resources term and instead parse it out into 4 different terms that kind of more explicitly call for different kinds of resources like materials, skills, time, or personnel. And then we've also added some additional terms as we looked at which terms are required through the OBPS for submission of their protocols. And so here, this includes what language the protocol is in, when the protocol was issued, a term about audience, which could be, is this written for scientists or managers, who published the protocol, and then if it has a version number associated with it. And so just going over kind of our themes for the week, yesterday we talked about the protocol templates and different efforts of harmonizing protocol. And today, we're focusing on the metadata. And so today, what our kind of key outputs that we would like to have is talk about this core set of standard MyOp terms, get feedback from you about them. But we also want to think about cases where a richer set of terms are needed for different types of projects or protocols. And these kind of will result in a list of issues that we can use on GitHub to track these changes. But just to start the discussion off for today, I think I'd like to discuss if you feel it's clear what we're doing with MyOp is trying to achieve. And can you see a benefit of having these terms for your work or other stakeholders' work? So I'll just stop sharing briefly here. And I can put that question in the chat too.
So this looks cool, Katie. How does it relate to the other MIXS standards? I guess like MIMARCs or MIGs or et cetera.
I don't know if Pierre Race, if you'd like to chime in on that too, but I guess, so MIXS is minimum information for any sequence. So that would be terms describing the sequences that you're submitting. And so I think there are some similarities in what we're asking people to give about protocols, but it's a slightly different goal, I guess. Pierre Race, do you have any thoughts about how they relate?
So, MyOp would be a checklist in itself. It would relate to MIGS in that it's also part of the Genomic Standards Consortium endorsed checklists. And then you can use that in combination with the MIXS, MyMarks, whatever other checklists that you need, and also whatever other environmental packages you need. And then MyOp is really very much only focused on the metadata of the protocol.
Yeah, just in the to highlight. Let's see, long time.
Hey.
Last time was Hawaii, I think that was fun. But let's see. Yeah, I linked into a line to show that we do import MIXS, right? So if we have to reuse a term, we're really reusing it. We're not duplicating it in MyOp. Like for example, which environment can use this protocol in, right? Is this valid in the Arctic or etc. So that's how it works. We're coordinated with the GSC. We're using their same LinkML YAML as Katie has introduced way, so that when we're ready to then launch this, It's completely compatible with their technology. But as Katie and Raisa has said, this minimal information is about protocols, not sequences, not the data sets, but really the protocols themselves. That's the thinking.
Maybe with that, another addition to what Pierre mentioned with the import of some of the mixes terms, we started with that. And then when looking at the definitions of, for example, the environmental medium for mixes, it really is, the definition includes where this sample was taken. And so those terms still need to be adapted to then be appropriate for the protocols. So we're looking at the mixes terms. However, because we saw that there was a clear distinction in the definition, we now refrain from importing them directly, just so that there's no confusion when looking at the YAML file.
Cool.
And I guess kind of another linked question that we could discuss at the same time is, do you think anything's missing in this set of terms? I actually created a Jamboard, which I don't know if has everyone used a Jamboard before, but it's just a way of us to also kind of spitball ideas as we're talking about this. Like even if if you have an idea and want to throw up idea as a sticky note on the first page where we have the MyOp terms listed out. If you think a term is missing or a term is unnecessary, feel free to just throw it on there too. So one of our major goals too with MyOp was, it's hard to decide which protocol you should use. And so we want to also make it easier to see, where protocols have been used before. So if you're planning on working in an area, you could maybe sort by protocols that have been already applied in that area. A lot of these are to kind of make discovery of protocols easier.
Nice, you shared your DFO guidance and your reporting templates. So I guess some of the fields in there you would consider minimal to find your protocols, right? So are there any that you think should be inside this list?
Yeah, I have to review what you have done before commenting, but I think it was great to put it there. So basically, we in our goal was not so much for scientists, was more like to compile all of the scientists from the lab and what the manager needs to have to make sure we can interpret the results. So there might be a bit different. At the end of the day, that's all information needed, but it's maybe the wording. I wonder if it's different. So I think it would be a good exercise for me to go one by one and look how, because some wording for us as a scientist or expert, it's quite easy, but for some managers who is not an eDNA expert but needs to make a management decision, I wonder how much it's easy for them to interpret those information. But I will do my homework and look at to link it. But I think a good things that we have started, it's to do glossary, because we are trying to have some kind of minimal data sometime that so that would be available on the website, public website. However, we know that some information might be sensitive because it's not published yet or something like that. So there's a link with the API, and when we do that, we need to make sure when we shrink it and make it like a minimal data and easy access, we still need all the complex data. So we did a glossary to make sure that we do the link between OBPS, mini DNA, and those type of things. And they have different wording and some really more scientific with very more details in it, whereas some others are more address different client or users. So I think it's quite important to have all of those level of definition.
Right, and I think in Oh, sorry, go ahead, Pierre.
Oh, sorry. But it's like just listening to that, I think that's already something that we could add. You described, Denise, the sense, some sort of sensitivity. So I put a sticky note on our Jamboard over here. That's something that we don't have as a minimal requirement, but maybe we should think about it. If there's some kind of sensitivity about this protocol, so are you using hazardous chemicals, radiation, but sensitivity could also be ethical. You know, is it like an indigenous area or something of that kind? Some way to sort of flag up front that there could be, that you must pay special attention to something in that protocol. An idea. So yeah, if you have others that you think should be in this, again, this minimal set, what everyone should be reporting about their protocols to exchange data about them. Please do add like sticky notes or whatever you want to the Jam.
One thing we decided to do was to, in the minimal, so you have like the GPS point, the time, and the eDNA concentration. So it's very minimal. And then when you can have an API which you have all the full data sets, fine. But if it's not published, or there's for some reason, you know, the author doesn't want to have all of the information in those meta-analysis, we always add the contact information. So that can be an API number, like public data, but that can also be someone that is happy to show the trends of a species of eDNA, but doesn't necessarily, he wants to publish first, like, you know, like, or any type of sensitivity. It's just trying to ensure that people are sharing data as soon as possible instead of waiting, because we know before it's all compiled, it can take a long time, sometimes a couple of years and things like that. So that's what, for the moment, we're trying to at least having contact information always, so, or a link to the full public metadata.
That almost brings up, like, you know, a maturity term, which I think we had kind of talked about before, of kind of what stage of maturity this protocol is in. We talk about sharing protocols early. That might encourage people to the sharing of early protocols if you say this is still under testing or something, but this is what we're doing right now.
Yeah, and that converges with some of the OBPSs on metadata, right? So inside the OBPS metadata spec, you have things like, well, the kind of protocol is it? You know, is it a SOP? Is it a recommended practice, etc.? So that's an option. Also, we don't know if that's minimal yet for the Omics community, but that is certainly an option. I'll put a link into that metadata spec. Oh, I see Pauline is also here, so she can cross-check that.
And if you want, I could share my screen for the Jamboard. Do you think that would be useful for those who can't? Maybe that's a good idea. Okay.
Yeah, also on the maturity comment, I think in one of the previous calls of the task group, we were thinking about using technology readiness levels. And in that, we're very aware that those are differently defined depending on what community you talk to, and would likely have to be adapted for the protocol case. But that might be one way of capturing the maturity of a protocol, that where there's a scale where people really know, okay, this is what that maturity level means. I'll drop a link to the issue on our issue tracker that is regarding that in the chat.
I see someone added a note. Is there a logic to the different ways multi-word fields are rendered? Words separated by spaces, underscores, and camel case. I think we have that multi-term category that we say yes, and then so can have multiple words, but I don't know if Pierre and Reza, you have thoughts of requiring certain. I think we just had it as strings right now, but.
Yeah, so we actually asked for guidance from the GSC, like, because Mix is also going through that sort of formalization and regularization of their fields. They also have some inconsistencies because they transitioned from a primarily academic group now towards a more formal standards organization. They just got legal status actually now. So they're cleaning that up. And when they clean it up and they define things, I think we'll follow suit. So we want to be compatible with the Genomic Standards Consortium specifications. Is that right, Raisa? Has there been updates?
Yes, yeah, exactly. And the two separators, I think, that were currently in question were either pipe or semicolons. And I know that Darwin Core, so another metadata standard that is very relevant for biodiversity data, they usually use pipes for multi-value fields. So that might be one way of also getting those two standards closer together and make it easier to move from one to the other.
Okay. Yeah, that was
Holly,
my
you have your.
question.
Oh, sorry. Go ahead.
Sorry, just to follow up. Yeah, it was more about the names of the fields. So like methodology category is just has a space, whereas has a version, has camel case, and personnel required has an underscore.
Okay.
It's just
a good point.
a little awkward. I mean, you could maybe just go with underscore since that's what GSC is doing now. But I understand Pierre's comment about like sort of updating that, like Darwin Core or EML. I think.
And there's
camel case.
there's also like the reuse thing. So like some of those fields are coming in from outside. They're coming in from Dublin Core or something, right? So we'd rather reuse them than try to create our own within GSC. And they may have a different syntactic convention, and we just go with it, right? Now, whatever user interface or so that uses them, you can clean it up and you can display it how you want to, but we don't want to create something just because we don't like the case. We want to reuse something that's really popularly used by libraries and others. I think that's where you were going.
On that note, so like broad scale environmental context and local environmental context and environmental medium, those are ENVO terms, but those have special names in MixS that have like they're spelled differently. So like env underscore broad underscore scale.
So those are the short names, right? So we didn't use the short names in this list for this slide because it's easy to read this way. But in the actual YAML spec, you'll see the names that are there. And as Raisa said, also, they're different from the mix fields because they're not the environment of the sample. They're the environment in which the protocol can be used, right? So there's a slight semantic variation, but then we're taking care of that. But this is the sort of thing. So if we read through this, it's like, okay, what category is your methodology? Is it sampling? Is it DNA extraction? Okay, give us information about your project. What was your purpose so that people can actually quickly search through the protocols? Give me everyone who's looking for invasive species, all protocols looking for invasive species, that sort of thing. Analysis used, etc., which environments, you read it that way. So then are we forgetting something? Is there something missing? Or we see some hands. I'll hand back to Kate.
Holly, would you like to go first? I know.
Sure, thanks. Holly Bowers, Montesani Marine Lab. Sorry, I've been lurking in the background for the past day. Kind of a different topic related to this, and I think it came up yesterday. Is there any way to link to information about alternative things that have been tried? So I'm just looking like, say, materials required. So you have that in the protocol, and perhaps somebody tried, Sterivex when it wasn't part of the protocol, and they found that it didn't work. And I think this came up yesterday about knowing the things, so we're not reinventing the wheel, repeating mistakes, and using up funding.
That's a really good point. And I think there you could almost link to, or was your point, something else was tried and it didn't work? Because that would almost like, if you had a different protocol of what exactly was tried and then showed the difference to say like, this protocol is linked to that one, this one failed or something. And that's when we made the change to the new one. But yeah, that's a great point.
And in a sense, like that would actually be in the Bebop GitHub architecture, because you'd have a protocol, you could fork it, right? So you copy it with the whole trace that it was linked and derived from something, all machine readable. And then you can say, okay, we varied these parameters and it failed. Maybe the MyOp field there is something like outcome, like outcome, like success failure, right? So the actual metadata field would be something of that kind. Maybe we'd have different levels, but we'd start with success, failure, optimal, suboptimal, use only if necessary, that sort of thing. But that's very useful. That's a very, very useful search vector. Like what has worked in this region and what hasn't worked in this region. And that gets us into that conversation we had yesterday.
Yeah, I think that's a great point, actually, because we've been talking so much about also wanting to share failed practices or all of the things that don't work. So with that would already present a great search factor for anyone who's looking into that. Thanks for bringing it up.
Oh, Frank, sorry, you have your hand up.
Yeah, hi. Thanks. And I may ask what may appear like a dumb question, but I was wondering, because I've looked maybe superficially at MixS, and I'm trying to understand, where do you capture very fundamental things like position and the format in which you code the latitude and longitude, or depth and time or date? Because that's always such an issue with people recording that in different formats. And I was trying to look at the MixS, and it says, well, depth is a field, but it doesn't give any guidance on units or any sense of precision of the depth sensor, or, you know, especially if it's positive or negative. You know, I wonder, and same thing for latitude and longitude. It says, will record latitude and longitude, but it doesn't tell you, though it says in decimal degrees, but it doesn't tell you much more, whether, you know, it's 360 degrees or negative, positive, which way. And so I wonder, where is that information? Is, if somebody wanted to document a protocol that goes with a sample, what, I agree with you that you have to capture a minimum of information, yet it's spread out in several different metadata protocols.
Yeah, so you're talking about the syntax, right, Frank? Like, what is the correct reporting syntax for latitude, longitude, units, etc. So again, as I mentioned, you know, Mix and the GSC came out of a primarily academic group that realized we need to get this data in order because things are catastrophic. So then they got organized and they produced these things. The first version was in Excel spreadsheets because that's what they knew how to do, right? But that already helped things. Now they've moved on to more like serious data management approaches, like that YAML repository, etc. And in the latest version of Mix, I think it's 6 right now, they're getting a bit more specific about that. So inside the GSC compliance and interoperability group, the SIG, right? This is where we're like sorting this out. There's also a technical working group where we're sorting this out, and we're saying, okay, we're going to fix the units so that we don't complicate the syntax. You know, there have been a whole bunch of recommendations, and in the next version, it'll be implemented. For MyOp, we don't have to wait. We can do it correctly, so to speak, right off the bat, and say, okay, here are the units for each one. But you brought, you know, the geographic location. I don't know if we've specified it. Let's like just take a look. And
currently, the geographic location doesn't actually pertain to latitude, longitude, but rather to, for example, if you get a sample at the Hawaiian Ocean time series, that you would know that it was from that sampling point, or at L4 close to Plymouth.
Right, so it's more like a gazetteering thing to tell us where you were. But that's actually a good idea. We could put in a minimal field, like if people have geospatial coordinates, they could put it in there in Wikit or something of that kind, GeoJSON. It's already a bit advanced from some, but we may as well put it in there. I'll put a note.
Yeah, I wonder. Oh, sorry, Frank.
No, go ahead.
I wonder if that was also part of why you brought that up. Do you think that latitude and longitude would be important, or would a more general geographic location be sufficient for protocols? Because I would imagine for samples, yes, you require exactly the latitude, longitude, and you need that very exact for where the sample was taken. But the question is, do we need that for protocols, or would that be rather not necessarily minimal anymore? But
well, to me, in my simplistic way, if I have a sample and I get a data point, that data point should have metadata that includes the protocol. So, and that is a protocol in itself, you know? So there may be nested protocols the protocol itself may be documented without a latitude and longitude, but it should include the requirement that if you're going to collect a sample, you should know where and when, because that's the only thing that's going to tie different databases together, is that exact location and exact time, including the depth. If we don't have that, we will never match one data with the other, except in a, synthetic way. But yeah.
Yeah, that's a good, I think we currently I know that Katie showed how we have the different stages that you have in Omics workflow, and part of that is the data management. And I think that would currently be where you would say, okay, these are the minimal metadata when collecting the sample that you have to collect and record and then share. But yeah, sorry, Katie.
Oh no, I think that's a good discussion. I was just going to say, Anais, I know you have your hand up for a while. Are you have a comment on the same topic or bringing up something else now?
It was for the topic before, so that might be a bit
okay.
the topic. We can come back after.
Yeah.
You want me to ask now? So basically, I wanted to know if, in terms of how it's reported, this mainly related to the question before regarding, which one is the best protocol versus the other and things like that. Do you have a decision tree that has been already done? Because I think that that's the best way to go now, where you have decision and things like that. Because that's all linked at the end of the day. Like you need to sample in a place where it's there's a lot of sediment in the water, so you're using a different approach in the field and then in the lab and things like that. So there are probably like some branches that will mix, but I was wondering how far are you getting to the best practices? I'm my perspective, it will be mainly a decision tree, so that's
right. So I think in the beginning we talked about the need and the want for a decision tree, and so I think this infrastructure is what we wanted it to do is help us get there, kind of. So what are the terms we need to kind of create that decision tree? These terms we can pull out of different protocols to make the decision tree itself. So no, we haven't made a tree yet, but we're kind of trying to build the framework. I think, first we felt like the first step was to make very visible exactly what everybody's doing and organize it effectively. And then we can have a better discussion about what may be the best practices based on that information for different regions.
That's good, thanks. I was wondering if you have already tried, because I guess that can be quite expensive by a series of questions and algorithms. So for the, I think Melissa has replied, we're working together in terms of the bioregion. That might be a good way to do if we are talking about minimal. In our case, that's what we are trying to do. Melissa and I is really to have the GPS point, but you can visualizing data and get access to the data by bioregions, because we're interesting how it change between regions all around the world, and if you are efficient, if you have it's especially about the optimal detection period. So we're wondering if the same species in cold water in the Arctic will be, or the same genus or something in the Arctic will be different than Australia and the Arctic versus a very hot country. So,
right.
Can I make a quick comment on that? Because the way that we have handled bioregions, there's many, from Longhurst to the one that we're doing now, like seascapes, which change all the time because they're based on dynamic satellite data. I would care more about the latitude and longitude and time, so I can position it, and then I would know if it's next to Hawaii or wherever. So if you give me some general thing, I'm in the North Atlantic somewhere, then I'm not so interested in that kind of a loose description of location, because I can figure out where it is if I have a good position. And I can put it in the context of a biogeographic region more precisely.
Yeah, definitely. For us, at least in our work, we have the GPS point, but when we can look about trends, where they have been detected and when, people can look about bioregion. So you compile data and see the difference. So it's some kind of a meta-analysis, but you can always have to go in the coordinate. I think it's crucial to go with the data. With the protocols, it's for us a different level where you basically have a project number, and that project number is then the same one for all of those symbols, so you don't repeating it. But the GPS point is based on the data on itself, and that's what we call the metadata related to protocols. So in our case,
one of our goals is to link these protocols to their data sets, especially with OBON. So I wonder if that can help try to make that linkage clear too. If we have lat longs for the samples taken with a certain protocol, then you could know, link to that data set, know exactly where it was applied.
Yeah, I think that's really nice to sketch it out in like why we do it that way. To say that, all right, group X developed this protocol and they tested it in the North Atlantic, right? This is where they developed the protocol and this is where they published it. And then there's a data set which says we used this protocol, but they used it in the Arctic or the Antarctic, right? So the GPS coordinate on the data set would be Antarctica, and the range of testing on the protocol would say it has been developed in the North Atlantic. That's interesting. It's just interesting information to see, where are the protocols being developed and tested versus where are they being applied when we get metadata from like the OBON spec. So that's a really good way to think of it.
Mm-hmm.
Yeah,
Jonathan,
sorry.
You have your hand up. Oh, sorry, Raisa, you want to go first on this topic?
Very briefly on that same note, I think it's really good to have that discussion on whether we should have the region or latitude, longitude, to be more specific. I think one of the things that we'll think about when taking this back to the development of MyOp will be how feasible it is to then provide that information for the people who are using the standard, because that's always something to keep in mind, that we don't want to put too much burden on anyone using it, which might be a way of moving away from latitude and longitude, because if you've tested it in 3 stations that are, or in 10 stations that are all within the same region, but not at the same latitude, longitude, then it's a lot easier to just put in the region. But I think we'll have a more detailed discussion on that afterwards, just to let the group know the different angles at which we're approaching the development of these terms.
Thanks, Raisa. That's a good point. Jonathan, would you like to share?
Yes. Okay, so I was just reflecting on the relative demand for precision for different data types and how that then affects the ability to determine quality of results between different studies. So geographic coordinates is one thing that's very important at the spatial scale, but going to the molecular approaches, the target, from what I've seen in the schema so far, it's focused just on the marker in a sort of general way and the taxonomic group. More and more in the literature, we're seeing how important just the choice of primers is for what you're getting, the TAC that you use, things like that. So I'm just wondering how detailed we might need to get with some of these particularly important variables in order to make the final data sets really useful for meta-analysis.
Thanks. That's a great point. Pierre, would you like to go first? Or?
Yeah, so like the MixS specification does have a lot of fields for that, which primers were used to generate the data, like the sequence itself, right? So, we don't want to convolute that, as you said yourself, the data set that comes out of it, right? We don't want to convolute that metadata with the metadata of the protocol. Now, inside the protocol itself, you would be defining which primers you use and a whole host of other things that are relevant. And that's also why we're using GitHub with this markdown format, because it's much easier and these templates, right? So it's much easier for machines to find out that information. The question here is, do you think that should be a top level minimum metadata field for the protocol? So you'd have like, because primers are not used with all omic protocols. There are primer-free methods, etc. Right?
Yeah.
So that's why we kind of rule it out, because it's not true of all protocols, right? So it's not minimal. What we would probably have actually is the same thing that happened to Mix. They have a core set like this, like Maya, and then they have packages and extensions for different kinds of environments, for different kinds of things, right? So we'd likely then have one for marker genes and stuff like that, an extension on this core that applies to all protocols. So we can pencil that in there. Please feel free to put in a note there. And maybe that's what we'll do next. But first we want to get Maya launched and functional, so that then we iteratively develop.
I think it would be a great idea to have, for example, a set specific to metabarcoding, protocols that I'm working on a lot with eDNA. So you can easily know which metadata key fields might be important among those studies to compare between lab protocols. But that would be a great idea. Kelly, would you like to share?
Yeah, now I have a lot of thoughts going on in my head. Okay, a couple of things I think are good points, which are instead of replicating what's in MixS or OBIS or Darwin Core, a requirement is that any data that you're using to demonstrate the efficacy of the protocol follows one of those, right? So that it ends up ingested. And I think probably more importantly for the protocol thing we're trying to do is how most people are going to be most interested in whether the protocol worked or not. And so I would like us to see, what's the new ground? What is, the things like lat long and all that stuff is super important, but they're basically covered in other parts. What does this group, or what does this protocol minimum standards, what is different about it? What's unique? And one of the things might be, getting people like how well tested, that you guys talked about a little bit, transition levels and blah, 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 and where the data is utilized. Like, how do we, go get to the wheat from the chaff, which is already an issue on OBPS, because you can go in and even 10 protocols is too many for the average user, much less 35 or 50. And so one of the things I was thinking about is maybe we could do some trial runs after we're done with this like initial discussion. Why don't we either pick some protocols that already exist and pretend that they don't, like we're going to create them, but in 3 different categories or something, where we hit different categories. And one of the protocols I was thinking, and I don't know whether this is appropriate or not, but this comes back to what Frank was saying, which is how difficult it is. Oh, I'm going to use MixS. And then you get in there and you're like, huh? And I want to make something that also works for Darwin Core or OBIS. And one of the things I was thinking is a much needed protocol is actually a script that converts whatever data headers you have, scripts that actually translate your data. So instead of forcing people to enter their data in a particular thing, which so far I would say just doesn't work, it just doesn't work, is a script to translate. And if we could develop that as a protocol, and the reason that's an interesting trial run is it's not a lab protocol, it's a data protocol. So maybe we could have one like that, one that's a DNA extraction protocol, one that's a library prep protocol, and maybe we could run it through and do some beta testing. Because what I found with Ocean Sampling Day and stuff, when we were all sitting here like this, it was like, and we need this, and we need this, and we need this. And then we were out there, particularly with the citizen scientists, and it was like, what idiot thought we needed all these things? That was me when I was sitting in my chair, and it was totally different if you're out there doing citizen science or you're at sea and, it's 30-foot waves and you're like, oh, forget you guys, I'm just trying to survive out here. So beta testing, maybe we could think of a beta test earlier, not later, an internal beta test on what we think our minimum that's that in a nutshell. Hope that made sense.
I just wanted to respond quickly to Kelly. I think the script is a good idea, but I think with the markdown formats that that Katie and you guys have started, should be possible to follow that template. And then so a script maybe to validate would be cool. That tells you, oh, you need to fix this, otherwise it won't be, you know, comparable to other people. I think it'd be hard to write a script to convert unless it was yet another template in like Excel format or something, but that seems like overly complicated.
I think to the point of like beta testing, I think that's what we want to do within the OBON program is here's a limited number of projects so far. I mean, they're growing every day, but or they're growing through time, but here's a limited group of people we could try to get everyone's protocols together in the same format and really have a test case and show how powerful that can be to then compare the data and map what everybody's doing and where. But also, I guess, to get back to capturing different types of protocols too, like, for example, I've been starting to put Ambari's protocols on the system and I've been concentrating on the lab protocols. So it is true that, you know, because I already have those written up. So we do need more different types of protocols to be put into the system as well to test it works for those as well.
Yeah, maybe that would actually be an invitation to anyone on this call who would be interested to try either the metadata specification or the templates, especially if you have protocols on, I don't know, on getting the permits before going out to sample, on how you manage the data. If you have anything like that already written up, which oftentimes I feel is only something that you talk about, but not necessarily have written down in a structured format, that would be incredibly valuable for us to get a sense of how best to structure that. And also might be interesting for you to get your protocols out there in a format that is being used by a whole bunch more people than just your lab or your project.
Philip, I think you've had your hand up for some time.
Hi. So I was wondering if it would make sense to have my op term for regarding the Q standards for contamination. So what is used for assess and control technical contamination? If it was used negative controls or PCR replication or coverage threshold by itself, it depends on the Omics protocol used, right? Multiple regions or markers or a combination of any. I don't know if it at this level it would make sense or not.
The field would be sorry, so that would be like positive and negative controls, right? And let's think, is that, so there'd be two fields, one for positive control, one for negative control. And that's actually important as a search vector because many protocols were not designed with that in mind, right? It's, I think that's also good to sort of help the community say, you know, we care about this. We consider that minimal, that your protocol should have a positive and negative control section. Many do, many don't. Thoughts from others?
Yeah, we had, I think when we were first talking about this, we had talked about separate protocols for like quality control of the data or quality control across your study. But yeah, that's a good point. Anais, are you responding to this?
I think it's because it will be filled by different people there. So I think the easiest way to do it easy and accessible, and it's basically breaking down that for people that are being in the field versus people in the lab and things like that, and then you're visualizing. So basically someone's that is collecting the water samples don't have the same app to fill versus the same. So quickly you get all of those minimal information that can be probably bigger and more accurate if it's only 10 questions for each part of the people. They were really generally, I don't know much people that do all of the steps. Most of us, we do section, we work together, but the information, even for some of my project will be hard for me to fill all this information quickly. I will have to contact the lab managers to see what they have been done and things like that. So that's already limiting me to fill that. If I have a link or an app to say, oh, I need you to fill that, and it's already thinking for someone in the lab, would be so much easier versus the person in the field versus. So I think that's the, in my perspective, that's the easiest way to get accurate and quickly, and people will be, it's on the top of my head what I did yesterday, but it's definitely not the same for the postdoc using the data or the person in the lab, or so that's how I will definitely separate that as a different people filling that.
Yeah, that's a good point, trying to make sure the information is filled not at publication time when it has to be submitted somewhere, but as the work is going on, or as the samples are being collected.
I think it also raises the point that we're now getting into some terms that may be really important for certain stages of the workflow, but not important for others. So, for example, with mock communities, or having a control or not having a control. That would be really important for lab protocols, but not really for getting a permit or for how you publish your data. So maybe that would be something that we'll have to think about, whether we'll separate that out and then have certain terms that are mandatory for certain stages of the workflow. That would be a way to approach that, to still have the ability to use those as search vectors and have them very visible when finding a protocol and helping you to find a protocol, while at the same time not asking somebody who has a protocol on permitting to put in whether they have a control or not.
So for today, kind of the last question. Oh, sorry, do you want to go?
I just wanted to highlight again that make it easy, you know, a little app or something on the phone like that. It's definitely the key for the person in the lab, just filling it right there.
But so yeah, so the ease of use, I mean, we're doing the back-end engineering right now, right? That would be then used to develop apps or so. But getting it right with these specifications is what's going to help those app developers, you know, make sure that they're putting it. And the point that Raisa raised, I think, is a very good one. And also why the modules are so handy, because we can split up what is minimal for each chunk of the protocol, from like sampling to the data to the permit acquisition. That could reduce the fatigue of people, because they're not going to be confronted with this massive list of things to fill in, right? It's like, if you're dealing with this chunk, here are 10 fields, you know, it's doable, right? And it all adds up. And also you can send that to the person responsible for that chunk, you know, because there are often different people in the team who handle it.
So I just wanted to say that that kind of leads into the last question I had today really well, because I thought, you know, we're talking about our minimum set, but then we had also talked about if we should have potentially new terms that are specific to either different types of protocols, so permits or permission or experimental design, or, you know, like we were just talking about, specifically for metabar coding or for certain purposes, when you might want to have a very specific set of additional terms that are necessary and that really help you compare protocol to protocol. But we also had a suggestion to really quick, maybe before we move on, if we could take a photo of everyone in this session. I might just take a quick screenshot for me. It's just nice. We're almost at an hour now. So if people could turn on their cameras real quick, I'll just take a quick shot of everyone. All right, thanks so much. I'm gonna say 3, 2, 1. I'll make sure to get both screens of people. Awesome, thanks so much. So I wondered if for this part, if we wanted to break into smaller breakout rooms just for 10 minutes and people can work on different Jamboard topics for ideas of terms that can be used for in different settings. And so I think I might just break us out into 3 different rooms. real quick, and we'll just do it for 10 minutes. And just for, if you have a new use case, feel free to create a new Jamboard for what you'd like to talk about. And we can just come up with some ideas for terms that might be important under different use cases. Okay.
So Katie, are the slides 2 to 5 the ones prepared for that part on the Jamboard, or?
Yes, 2 through 5. And so the second one is about methodological data categories. So that is about the categories that we define for groups of different protocols. So yeah, as a breakout room, you can decide if you want to talk about those broad overall categories, or if you'd like to focus in on a use case where we might develop a certain additional set of terms that are important for that use case. I think I've got the 3 rooms set up, so hopefully it'll close in 10 minutes and bring us all back and we can report back. But I can also jump between rooms to check in on how everyone's doing. So see you back soon.
Hi guys. Okay, so we have the unfortunate situation of me being in the room and me recording. So everybody be careful what you say. But I think it should be fine.
Well, I mean, just to start it off,
Yeah.
I mean, I think it's important to remember that this is metadata about the protocol, right? So not about the data or the event, which are already well defined and would be captured, right? And I think based on the feedback we've gotten from queries and thinking about how to distribute your protocols, the feedback I always get is like, how much is this going to cost? How much expertise is it going to take? How many people? That's the kind of core stuff that like, and then they'll follow it, but just some really simple aspects to the, even if you compartmentalize the steps in our value chain that people want to know towards, like, can we even follow this protocol? Like, that's like the really important data that I think people, as they search across protocol space, want to be able to wean or weed their viability down into. Of course, what studies or whatever have been derived from that is valuable, but there's a really set of, like, just kind of functional metadata that is missing in the reporting of the data itself that I think we're trying to capture here, if that makes sense. So.
Yeah, great point, Chris. I'm wondering, so currently looking at the 3rd slide on the Jamboard, where we have all of the different terms that we currently have in MyOp. We have the materials required, skills required, time required, and personnel required. Would you suggest to add another term on funds required?
Well, I'm just thinking, like, materials required, I guess you could just look, it's like an ingredients list, like we talked about. It's like, can I make this meal? Do I have it in my cabinet? It's like, can I find it at the store? Like, like there's stuff I'd love to cook, but we just can't find Thai basil this time, or whatever it is. It's kind of the same principle. It's like, can, and then if I choose to substitute, is it going to invalidate, the capacity to compare? Because that might affect my decision to follow through, even though I'm kind of changing an ingredient. Is it significant in changing the flavor of the outcome such that, I no longer can draw that comparison? That's somewhere in this protocol metadata. That's the critical stuff I think we want to try to. So, like, one of the questions was, has this been cross-validated, for comparative purposes across minor substitutions or something like that? And that could get to the mock community discussion that was Pierre dropped in or whatever. Like, it's tricky though to capture all that, right? I mean, I think that's what we're struggling with right now. So. And I don't know if there is, like, can we literally borrow strategies from like cookbooks or from something else that does this? Like, you know, that people do this. It's like, you know, like if you don't have this, feel free to substitute in this, you know, like how do you capture that?
I feel like the last point might be more of a thing that you capture in the protocol itself than on the ingredients list there. That could be captured, but the point on the protocol having been cross-validated or anything like that, that might be an option for a new term to be added to MyOp, which may, I don't know, I'll have to check again. It may be actually covered in the technology readiness levels, which I'm not sure whether it is. I don't know, is anybody else familiar with those and just has them on the top of their head?
I thought there might be an endorsement category as one of the additional terms somewhere in the back of my mind.
Yeah.
But this would be different than an endorsement, right? Because you could think about
True.
like like we talked about piping multiple entries. So if you can imagine, it could be a long string of piped cross-validated ARCs or GUIDs to other protocols that are cross-validated or something like that, right? So it could say such that it builds a set within that string that could demonstrate or at least support cross-validation or something like that.
Oh, I see what you mean, across different protocols.
Yeah.
Right. Yeah.
And you could, much as like endorsement could be multiply endorsed, right? But what underpins that endorsement hopefully would be some sort of, well, somebody might just endorse it because they made it and they're like, we like this and we're sticking to it, so I endorse it, you know. So
yeah.
Yeah, but Jody, indeed, I think as part of the OBPS metadata, they have the endorsement process because they also have that conversation with GUS of having EOV or GUS endorsed protocols for the reporting of the EOVs. I don't think that we included them in MyOp because that would be a second step. So once you've put the protocol out there, that could be an addition, but not necessarily at the point. It may be also at the point of uploading it the first time. But yeah. I think
possible. Oh,
yeah.
Is it possible to have alternative categories? You have like just even, for instance, the location, I think is critical. Thinking of some methods are tested in a location, but if you don't validate it in your new location, you know, it's a different community or slightly different interspecific diversity, genetic diversity, that you may not be picking up what you think you're picking up. So I could see just even location, could you have like an alternative, you know, cell where you've got for location or for TAC, you know, and then that allows that to have multiple categories that have been validated. But I don't know how the framework goes, so it could get complicated.
I think, so what you're proposing is to, for each location where you've tested, well, for each location that you say that protocol has been successfully used, to then have an added field on that to say how it was tested, or what exactly?
Just like have multiple places to enter GPS coordinates, something like that. Like these have been validated here. And then the same thing, it's been validated on this TAC polymerase, it's been validated, you know, trying to get at Chris's adding these different ingredients that might be swapped out, or like I want to use this because I know it works in this setting.
But Maggie, how do you determine or validate success, right? Because like you're saying, if you can say, oh, I did it and it worked.
Yeah.
That doesn't necessarily mean it's.
But isn't that what all these do?
Yeah, well, that's what I'm trying to figure out. Like, how do we
clarify that it works? So that's where my question of controls, reporting your control, that's what I always go back to. Like if you don't report those, you can't tell quality or accuracy. So those might need to be incorporated somehow.
Yeah, that's a good point. Also, because I think looking back at the environmental broad scale and geographic location terms that we have, I think the way that we defined that was to say the environment in which the protocol has successfully been applied. but I don't think we specify what we mean by successfully. So that might be a good point to say, okay, successfully means that you've done this, this, and that. Because of course you can always mix things together and something comes out, but how do you know whether it's what is actually there?
So you almost need a minimum standards checklist. Like, did your protocol, did it do in situ, did, you know, all of those things that you want to validate to be able to make sure that, because the last thing you would want to do is put a protocol out there and have someone pick it up and then it's bad for some reason or other. And then it affects, you know, I'm just, managers are very untrustworthy right now. So that would be scary to me, but I guess it's up to the person and how well they, if, you know, if they want to put a bad protocol out there, then that's up to them. But I do see a lot, like being a journal editor. Wow. The stuff that people put out there, it's a little frightening. So.
Yeah, it's scary. Okay, I think we still have like 30 seconds, but we may get an additional 60 seconds afterwards. I think that's usually how Zoom does it. So any other terms that anybody has been thinking about that we maybe should be adding? Okay,
I guess I was curious. Oh, you had assays, but then also like, will it be captured? Like if you want to go digital, quantitative, CRISPR, will that be captured somewhere? And then additionally to, is this an extraction protocol? Is this a filtration protocol? Maybe those don't always matter, but it might be nice to link those to some degree.
Yeah, so currently that would be captured through the step of the workflow that it's in. So you have a sampling protocol, a DNA extraction protocol, a PCR protocol. So I think we do have that in that sense, but, oh, okay, yeah, still 60 seconds. So that way we could have that captured, which links back to whether those different steps in the workflow are sufficient that we have at the moment, but maybe that's another discussion.
Yeah, and sometimes you want to do it a certain way for a certain technique, you know, so it depends, like those workflows might need to be connected. Do they have connections? So like this isolation is really good for metabarcoding or this TAC, that kind of thing.
Yeah, so the thought was that we'd have the different value chain broad steps, and then you could string them together as a linked set of protocols. So, you know, kind of like building blocks that would say, you know, these are, and therefore you develop these canalized systems that are consistently working. And so that would demonstrate, you know, that kind of
Meeting is being recorded.
Speaking of managing the WhatsApp, the technical WhatsApp, is there a way to adjust? Like I'm getting every single thread anytime anybody WhatsApp someone for technical help. Is that the way it's supposed to work?
Mute certain groups.
I can make the inquiry, Kelly. I'll find out if it's possible, but I think it's a service. And yes, I'm sorry about that. You could possibly remove yourself from the group, perhaps. I don't know quite everything about WhatsApp, but I will ask Ben, okay? And we'll get back to you.
Thanks. Wait, welcome back, everyone. I thought maybe we could all, one representative maybe from each breakout group could share what was discussed real quick. And I don't know if somebody else from the room that I was in wants to go instead of me, or or I'm happy to do it too. I can just say, okay, so we kind of concentrated on permits and permission protocols. So basically we started brainstorming what are the different things you have to take into account when you're sharing those types of protocols for either, you know, dealing with the Nagoya protocol or like permission to sample in a certain region. And I think we kind of thought it was or I think at least it's a hard issue. Like we thought of a lot of different considerations that have to be in place. It depends, you know, who you are. Like, are you part of some agency that already has blanket permission to work in an area? depends, you know, we kind of came up with some terms that we thought would be important to help the next person. For example, contact information, what agency or person was contacted to get permission or permitting, kind of if there's a cost involved, what's the purpose of the samples that you're collecting, and what type of substrate are you collecting, and are you in a marine protective area? So I think there's a lot of additional terms that might be needed when we think about those types of protocols that might help those who want to figure out how to sample in a certain area. I don't know if anybody wants to add anything else to that from my group. We were on slide 3 too, if you want to take a peek in the Jamboard. I guess would someone from another group like to share real quick what they focused on in their discussion?
Yeah, sure. So like in our group, we took a look at, we bounced back to the average, the overall minimum metadata. And it was very like the idea if a protocol was derived from another one, there was already a sticky note up there saying, you know, relationship to other protocols, like does it mix them, etc. But I mean, having that sort of derivation is important. If the protocols were developed in GitHub, that's not a problem. We can trace it with the repo metadata, but we have to recognize that a whole bunch of them can be coming from who knows where, right? So the ability to say that this protocol came from another one is important. We also worked on another board we added, is number 5, I think. It's and for bioinformatic protocols, right? So the bioinformatics or in silico analysis chunk. And we just thought about okay, what the minimum metadata there would be for that module. And that things includes like the links to the software, the links to the code in the DOI sort of thing, any example data sets and validation data sets you can use to make sure the stuff is working, information about the hardware required to run the bioinformatic protocol. Do you need a cluster? Do you need a simple laptop, etc.? And then many of the others will overlap with the main one. So we just brainstormed a few there. And yeah, again, this picking up on this idea of having a set of terms per, a set of MyOp terms per module, per thematic module or methodology category.
Mm.
Yeah, I think that's, sorry, just a response to that. I think that's interesting, especially with the in situ metadata and the template, because I think there will be a lot of overlap between how we currently have structured the template for any in silico, I meant, work that you do, and then having the link out to the protocol, having an example data set or anything like that. So that it would be great if we could somehow populate part of, either based on the metadata, populate part of the protocol, or based on the protocol, populate part of the metadata, to avoid people having to put it in each time, especially if we want to make it more specific for module in the workflow. Because the more specific we get, the more overlap I think there will be between what you put into the protocol template and what you put into the metadata fields.
Yeah, I think
that's a great point.
I think that if the templates are stable enough, we could certainly write a GitHub action or a script to auto-populate then the metadata. But that means that people can't change them, and we have to think about that in terms of implementation, how we want to approach it. Because what we will need is a stable set of MyOp terms in order to do the auto conversion to JSON and then feed it forward into the whole Decade digital ecosystem thing. So we need some stability at some point.
I think in some cases, like for example, a PCR protocol, you could have a table that remains very stable with your primer sequence and, and that sort of thing. So some of those may be able to just remain stable and be automatically pulled into some kind of metadata.
Yeah,
category.
and also with the resources table, I think in most of the templates we currently have that. So that could also be a way to automatically populate the terms and materials required. I mean, which, yeah, not necessarily. I think then in the in silico protocol, that would be what, yeah, what hardware, what software you need, whatever.
Would someone from the last group like to share too what they talked about?
I could just go ahead, and then anybody from the group, feel free to jump in for more detail, or if I missed anything. So we looked just in general at the terms that we have at the moment, and looked a bit whether we can think of some that are still missing. And I think so one point that came up was about how to capture, or whether to capture, that a protocol has been cross-validated. And that then led us to, I think, in the terms that we have, taking inspiration from mixes with the environmental broad scale environment and the medium scale environment. We currently have in the definition that is the environment that the protocol has successfully been applied in. And so that got us on the question of what does successfully really mean? Because you can always mix a bunch of chemicals together, something comes out, but how do you know whether that's representative of what's actually in the environment? So that then linked to the cross-validation, again, to having controls, anything like that, which I think will be an important discussion to maybe have, also following after this workshop. Sorry, just got a message. Following after the workshop, to specify that and maybe say, okay, these are the things that we expect when you say something has been successfully applied. And then the last point I think that was mentioned was also about stringing protocols together, not necessarily what Pierre mentioned with the derived, that you have a version that comes from something else and was adapted, but rather to say this protocol works really well with that other protocol module. So if you use this extraction method, that's the one that works really well afterwards. So again, to highlight that there is a need to be able to link those protocols together and really make use of having them in modules, which I think we've talked about before, but I think was also important that came up again. And yeah,
that's a great reminder.
Yeah, we talked about it before, but in terms of implementing it, I think, yeah, if people are using modular templates and they say, okay, the output of this is fine as input of something else, I think in Bebop we would have like some guidance on how to create a workflow based on those modules. To say, fork the various modules, tie them together, and this is the template for a workflow. Now that actually fits into Obon's 10-year strategy, right? Because by the end of it, we want, let's say, machine actionable plan. So a machine would be able to say, I need this module, this module, and this module. Can I auto execute them? And there are these like common workflow languages and machine readable directives. So this is a small step in that direction, but a very important one. Just getting us humans used to the idea of being a little bit more organized and structured with how we talk about protocols.
I think we had also talked about it, how you could, look at a data set and then see the exact string of protocols that were all used in order to generate that data set. So I think that's also very powerful
exactly,
too.
Because then you don't need like a whole series of DOIs with all of the modules. You have one DOI that points to the workflow that does the stringing by itself, right? So that actually saves a lot of overhead in terms of data transfer. We'll think about that. Maybe we should, I'll create an issue on our Bebop tracker just as a general thing.
Aeneas, you have your hand up?
I think it's very important. If it's done, sequentially, so you have the modules and people feel it, a lot of people will tend to say quickly, it didn't work. Basically, project that has not been successful, people will not spend their time to fill the forms and fill all the data. So if it has been done, previously, how it has assembling design has been done, collection, and then 0, values, it's still valid to learn for the best practices. So for those type of things, I think that will help personally to get the people to publish those unsuccessful, so we can learn from this.
Well, I think we've got 10 minutes left. So I don't know if anyone wants to bring up anything else before we have our end of our discussion. But I think we talked about a lot of really interesting points here today that we'll work on GitHub. And just to say again, we meet about every other week to talk about these issues and to work on them on GitHub. And please, you can contact me. I'll put my email in the chat if you'd like to join our meetings, or if you'd like to just contribute on GitHub too, with issues or different things you think would be good ideas to change. Or if you'd like to, please, we're looking for more people also who want to be early adopters of the protocol templates and add their protocols to our system too. So
yeah,
that would be great as well.
Just to underscore that, if you do add protocols to that system, you're directly plugged into OBON deliverables, and it's very easy to get that recognized as a decade contribution. If you don't want to apply for a whole project or something, even just the fact of getting your protocols into the Bebop system is recognized as a contribution to OBON, and it will also help us make sure that when we develop the system, it will be suitable for your needs, right? Because we can only work with what our projects and our members and contributors are telling us in OBON. And of course, we will do the extra work of making sure that the metadata, et cetera, gets federated out to the rest of the UN Ocean Decade system. So, we add that value to you and provide that for you.
And also, just as a reminder too, we're meeting once again tomorrow, same time. And Aeneas will be leading us tomorrow in a discussion about kind of focusing more on sampling design and concerns for sampling. But I don't know, Aeneas, if you want to say something real quick about tomorrow.
Yeah, we'll try to basically focus mainly on sampling design. When you start a survey, how do you start it? Where do you find the informations, considering the biological and physical factors? If you're trying to starting by pilot project, or you typically do literature review, do you feel confident on applying those protocols in your regions, or how, like, just getting a better understanding on how people develop their sampling design, and then how do you interpret your results? If you have been a bit like randomly and then you get successful, are you confident to compare data all the time? So what is missing there to get those confidence? And it'll be very interesting. Fun discussion. And we'll continue. If you are thinking about questions from today, that's definitely a good way to, we can address that and go forward with this.
Thanks. Well, thanks everyone. Thanks for those of you who are contributing early or very late. I think there's one hand up. I don't know if Yiming, if you'd like to say something.
Yes, I put my hand up just in time. So I have a question regarding this MyOp. Is there any intention of producing something like the Darwin core mixes thing? I guess Raisa would understand what I meant.
Yeah. Raisa, would you like to respond?
I'm sorry, my brain's not working anymore.
I know, it's way too late, isn't it? But yeah, so we with the task group under GIBWIC, so that's a joint task group from TEDWIC and the GSC. We brought together some of the developers of the standards, so the Mixes standard and the Darwin core standard, as well as adopters. So we had representatives from OBIS. We also had Yiming joining to develop a mapping between Mixes and Darwin core, because both of those standards are so relevant for omics biodiversity data. And with before that mapping, we're still creating data silos when you use one or the other, because they just didn't work together. But yeah, that's the background. That's actually a good question. I didn't think that we plan to do a mapping from MyOp to Darwin core, because there should be some overlap between Dublin core and MyOp, because we are reusing some Dublin core terms in MyOp, but we're doing that by also then reusing the IRI that was issued for the Dublin core term. So it's not that we're duplicating it, but rather that we're importing that term in a sense. But then at the same time, as we mentioned for some of the Mixes terms, we took inspiration from the Mixes terms, but adapted them because the definition didn't quite work for what we're doing with the protocols. So I don't know. Currently, we're mostly focusing on getting MyOp as like the core terms down. And then I think once we have that, we'll evaluate whether there's a need to map that to any other standards. But at the same time, while developing it, I think we're trying to avoid needing to do a mapping then in the second step. But if anybody else on the groups wants to jump in, please do.
Yeah, I think that's right. A lot of the basic publishing metadata will be covered by Dublin Core, so that's already in Darwin Core specs, right? We can also use the extended measurement or fact part, for all of the extra stuff, which for sure isn't in Darwin Core. Like a lot of these things are far too detailed. We can use the IRIs that the GSC will provide for the MyOp specification in the extended measurement of fact. As in the Ghibli group that I linked to, there's our final report, which explains our recommendations for this. And I think that's how we would do it. We'll talk, of course, to Tadwig to make sure that this works for them, and get the endorsement of the executive. Raisa is also part of the, was it the technical work, the tag, right? The technical.
Yeah, the Tadwig tag.
Yeah. So we're going to make sure that this is like legit in the sense that it's not just like a random recommendation. It passes through the standards bodies. We recently got the executive of Tadwig and the GSC board to agree on a memorandum of understanding for that mapping from Ghibli. And I think we can do the same process, but as Raisa said, after we have some stability and we're sure like, okay, this is good, this is community vetted, now we can bother the people on those various panels and boards.
That's awesome. Thank you very much.
I feel very lucky to have Risa and Pierre working on this project too. Well, thanks everyone for a great discussion today. And hopefully we'll see some of you tomorrow too for our last session. But take care and thank you.
Thanks, Katie.
Thanks, Katie.
Thanks all.
Bye.
Yeah.
Thank you.
Thank you. Bye, everyone.
Thank you.